Hi, today we're going to go off in a slightly different direction from previously, and we're going to be talking about DNA and genes. I apologise if you've heard about this before, or maybe even know a lot about it, but the reason we're covering it in this course is so that we can understand the process of the genetic origins of inheritance through natural selection. So we need to have a complete understanding of natural selection and genes, DNA and genes. So this will just be a brief touching on the subject, not too technical, not too extensive. We'll be talking about the human cell, the DNA, chromosomes. Get back to there tonight, and we'll be doing which is the structure of genes, and we'll be doing a brief history of the discovery of genes, and then we'll take a look at the modern science of evolution, which developed around about the 1940s and 50s and has been developing ever since. So this is an essential part of the course, and I apologise if you already know about it, but look on it as a very essential revision to bring you up to date. And also you'll know how I think about it, what my take on it is. So first we're going to be talking about the structure of genes, and we're going to be looking at what is a cell, what is DNA, what is a gene, what is a chromosome, and how many chromosomes do we have. And firstly, what is a cell? The cell is a basic building block of all living things. In other words, our bodies are made up of cells. All living things are made up of cells. These cells are descendants of a 3.5 billion year development of life on Earth. And we won't go into the details in this subject of how life developed. Essentially to say that it's been developing for 3.5 billion years, and for the first 3 billion years, it was mainly single cell creatures, and then later in the piece, the cells decided that they would clump together and have a sort of a federation of cells, and then they found that it would be more efficient if the cells not just clumped together, it became different parts of a whole body. So each cell, the cells started to specialize and play different organs in the body. And of course, that was happening over the last half billion years, 500 million years. And what we have now is where the end product, or the current end product, because we're not the end product in the big picture. In the big picture, life will go on evolving so long as planet Earth is still livable. And even if it isn't livable, a different form of life may emerge. But anyhow, we're at this point in evolution, we are sort of the product of all those cells working together. And like I said, they're the basic building block of all things. They provide the structure for the body. They take in the nutrients and they convert nutrients into energy. So they're very important little factories in our bodies. And they also contain our hereditary material, our DNA. And not only that, they make copies of themselves. And every time they make copies of themselves, they duplicate the hereditary material, the DNA, in the nucleus of the cell. And of course, the reason that they make copies of themselves is that cells don't last forever and they need to be replaced. And if you've ever noticed that sometimes if you take your socks off, you find you got little white stuff in there. And that little white stuff is probably, unless if you've got black socks, I wear black socks. That little bits of white stuff is dead cells that have decided that their time is up and they've been replaced by other cells. Now the DNA. So what is DNA? Well, its proper name, its full name is deoxyribonucleic acid. And it has been around for most of those 3.5 billion years. It started out as RNA, and eventually they developed into DNA. And RNA, of course, is just ribonic nucleic acid, and it's still around. We still have it, and it does serve a purpose, which we'll get into that later. It's the hereditary material in organisms. Nearly every cell has the same DNA. In other words, in the human being, in the body of the human beings, that DNA has been copied into each cell is the same in every cell in the body, apart from a few. And they're located in the DNA is located in the cell nucleus. That's the very center of the cell, which is a protected little compartment. And we also have a small amount of mitochondria, which contains the DNA. And mitochondria is the part of the cell that converts food energy into energy, into a form that the cell can use. So it's a very important part of our cells, and it contains some DNA. And you might have heard of female mitochondria being passed down, and that's how they trace the origins of people to a large extent, at least on the female side. Now, talking about DNA bases. Now, bases are like what do you call it? The bits of information, tiny bits of information, and they come in 4 flavors. We've got adenine, guanine, cytosine, and thymine, and they've been abbreviated to A, G, C, and T. So when you're looking at a genome, you'll see all the little bits of pieces, all the information pieces are labeled A, G, C, or T, and the combinations of that. And we have in our human DNA, we have 3 billion bases. In other words, these little bits, these chemical bits of chemical, are put together to combine in 3 billion bits of information, which is a lot of information to make a machine like us work. The base pairs are bits that join together, so you get a discrete piece of information. 2 bits of these A, G, C, and T bases join together, and they're called a nucleotide. Now, the DNA is arranged in a double helix sort of fashion. It looks a bit like a stepladder, a bent stepladder. You can imagine a very long stepladder that's been twisted. And the vertical parts of the stepladder are made of sugar phosphate molecules. And in between those, we have the rungs of the ladder, and the rungs of the ladder are the bases that we talked about just a minute ago. The base pairs are the rungs of the ladder. And every time a cell divides, the DNA replicates. So you get a complete, if your body's working the way it should, you get a complete copy of the DNA in each cell, so that each new cell has an exact copy of the DNA in the old cell. However, on the downside, there's a limited number of replications that a cell can make, and it's usually around about 50 to 55 replications. And if you live to be over 50 years of age, 55 years of age, then you begin to notice that your cells aren't like lovely and smooth and firm like they were before that. And the reason for that is that our cells have decided that it's not possible to divide anymore because they can't copy the whole DNA after that number of replications. And there's a reason for that, which we'll get into later. Now, genes are the basic physical and functional unit of heredity. And the gene is a section of that ladder, that strand of ladder. And in each gene controls the chemical and physical characteristics and behavioral characteristics, not each gene, but there is a gene or a combination of genes behind all our physical and chemical and behavioral characteristics. When it comes to the behavior part, though, we do have to have a caveat on that. And the caveat is that behavior is half genes and half environment. So we need certain things to happen in the environment to switch on the particular genes, the particular combination of genes involved in the behavior that's appropriate to that environment, or in some cases inappropriate to that environment. So it's not just it's not just instinctual. In other words, we're not working on pure instinct. We need the environment to switch on whatever it is that we've inherited. So each gene has up to 2 million bases, 2 million bits of information. So you can see there's plenty of knowledge in our blueprint. If you take the blueprint of a human being, it would, and laid it out, it'd probably cover the whole of the surface of the planet Earth. Yeah. And we human beings have between 20,000 and 25,000 genes. Before they actually discovered it, people were making estimates, and they put it in the hundreds of millions and the hundreds of thousands. And when they found out it was only 20 to 25,000, they couldn't believe it. It was like, that's not possible. How could you build a organism like a human being from that small number of genes? However, we have, and we do. And it's very clever the way it's done. We won't get into that today, but it's done through repurposing. You don't have a single gene for everything. Genes can do multiple jobs. Okay, and each person has 2 copies of each gene, one inherited from each parent, and we'll get into that in a minute, a bit more into that in a minute. Now, after we've inherited our 2 copies of DNA from our parents, one from the dad and one from the mum, each version, because they're 2 different versions, and they're not identical, the 2 different versions of each trait, be it a physical trait, a chemical trait, or a behavioral trait, is called an allele. And you can have a number of alleles side by side. Usually there's only 2 side by side, but I have heard that you can have more than that. And what that means is for any particular trait, be it physical, chemical, or behavioral, your system has the option of choosing one or other of those alleles, those traits, either from your parent, either from your mother or your father, or from your ancestors, and from your ancestors. And according to the theory of heredity, it's usually the dominant allele that will win and will take and provide the characteristics that particular person will have. And the reason that they are alternative, like they're not the same, is that there are small differences in the sequence of the bases. So the bases don't all come in the same order, same sequence, and when they're in a different sequence, then we get a different instructions for the body to build the mechanism. Okay, moving on. Next, we're going to look at a chromosome. What is a chromosome? Well, we've got all this DNA in there, However, might get a bit messy, and after all, these strands of DNA are very, very long, and they get neatly packaged into little packages called chromosomes, and these are inside the nucleus of each cell. And the chromosomes are made up of proteins and DNA organised into genes. Now, as I said, genes are a segment of the DNA ladder, the double helix. So it might be over a centimetre long. That's a segment. So all the information in there controls maybe a couple of your traits. And these are packaged into chromosomes and are coiled very, very tightly. Now, in the middle of the chromosome, you'll notice that sort of the chromosome joins, not always in the middle, sometimes it's in the middle. If it joins exactly in the middle of the two sides, you've got actually two strands of chromosome. If the chromosome joins in the middle, then that's an X chromosome, like that. If it joins the top or the bottom, then that's a Y chromosome. And the purpose of these are called centromeres. The purpose of these centromeres is to make sure that when the cell divides, it divides properly. So it has a very important role to play. And at the same time, it gives each chromosome its distinctive sort of label of an X chromosome or a Y chromosome. And you might have heard that males have an XY chromosome and females have an XX chromosome. Okay, let's put this one now. Let's get more accurate here. The gender or the sex chromosome in each of us, if it contains two X's, then it's a female. If it contains an X and a Y, it's a male. So we have 23 pairs of chromosomes in each cell. And like I said, the 23rd pair is the sex chromosome. Females have two X's and the males have an X and a Y, as I said before. And just briefly, yeah, we're getting onto it later. When a male and a female come together and the egg is fertilized, the female egg is fertilized by the male sperm, then you end up with 46 chromosomes. In other words, you've got 23 of your mothers and 23 of your fathers, and they fight for dominance. Okay, now we're going to look into, that's so much for the biology. Now we're going to look into the brief history of the discovery of genes. And the first chap on the list is a guy called Gregor Mendel, who was an Austrian Augustinian monk. I think in this day and age, he would be called a nerd. He was a mega nerd. And he spent almost his entire time in the monastery studying peas, the pea plants. And he's crossbreeding them, different varieties of peas, to see what were the rules of inheritance amongst peas. And he found that the heredity acted in a predictable manner, and that if you put a pea with a white flower together with a pea with a red flower, you don't get a pink flower in the offspring. You get either a red or a white. And he came up with the idea that we have dominant and recessive traits. Now, he didn't know about DNA at this stage. He just knew there was a mechanism in there that if your father has one trait and your mother has a slightly different trait, it's the one that is dominant, the one that is strongest, that will win over. But thinking along that line, you might say, well, why aren't all the brothers and sisters in the family look the same? I don't have the answer to that. No, they don't, because there's the way that our DNA can express itself is sort of infinite. there's almost an infinite variation among those 2 billion bases, or whatever, 2 million bases in a gene. That's right, 2 million bases in a gene. Then there's almost infinite possibility of combination there, within the limitations of the organism, of course. Now, Gregor Mendel, so going back again. Yeah, I've got a bit out of order there. Okay, skipping down. Now, his hereditary, his laws of heredity were that we get two elements. You call them elements, but today we'd call them alleles. They're segments of DNA. We get one set from our mother and one set from our father. The dominant ones win, and a single allele can control more than one trait. In other words, as I said before, there's a lot of efficiency in the body. We don't need one gene for everything. No, for one gene per everything. We have a combination of, a gene can do a combination of jobs, and they do them in combination with other genes as well. So that's his laws. You get one set from your mother, one set from your father, and the dominant one wins. You don't end up with a mixture of both. Okay, now moving on. There are exceptions to the law, of course, that I don't know whether he outlined them, but we certainly have. Many plants can reproduce asexually, so they are actually clones of each other. They don't have any variety, any variation, not from receiving a different set from each parent. So incomplete dominance of one allele over the other. So in other words, if you get a situation where one allele is not so dominant that it can overpower the other one, then you get a bit of a standoff there. And co-dominance of alleles, in other words, they're both dominant, so that's the opposite, where they're both powerful. And polygenic traits controlled by more than one gene. So what they're saying is that like I said, you don't necessarily have one gene for each trait. You can have a bunch of genes for a trait, and they may not be the same in every organism. Right, now the next guy on the list, this is 1910, was Thomas Hunt Morgan, and he worked on fruit flies, not peas. So he went up a bit. I guess one of the reasons that they work on fruit flies is it's easier to get ethics approval for the experiment. Number two, the fruit fly has a very short lifespan. In other words, it goes from generation to generation to generation to generation really quickly. So if you're studying evolution, then you're seeing natural selection in process in the course of one day. Whereas if you studied human beings, you'd have to look at a period of about 100,000 years. So he discovered that each gene is located in a chromosome in every cell, which we've just been talking about. And single genes alone cannot explain inheritance. So in other words, it's not down to single. We don't have single genes for anything. Everything is a combination of different genes. So if someone tells you you have a gene for this and a gene for that and a gene for the other, they're not speaking the exact truth. The truth is you have a combination of genes for this trait and a combination of genes for the other trait, and some of the genes for that trait might be involved in the genes for this trait. So it's not as simple as people would like to have it. And of course, he discovered the genes were packaged in chromosomes, as we've been talking about. And each individual has 2 copies of each chromosome, one from their father and one from their mother. And humans have 23 pairs and each, but when you come together, you have 46 in total. So we actually have 46, 23 from our mother, 23 from our father, as we said before. But this is the guy who discovered this. And as we said, he discovered that we have 2 copies of each gene. Chromosomes come in pairs. Each individual has 2 copies of each gene. One of each chromosome, and chromosomes become intertwined. Now, that's very important for mutating and creating something out of the box, something different. and it's a source of genetic variation in natural selection. In other words, when chromosomes become intertwined, you get a thing called recombination. In other words, bits, it's a bit like a bit of the information in one part of one chromosome is exchanged with the other chromosome, and it might be put in a different place. And quite change the trait a little bit, or change it so much that life is not possible. So that's a something that adds weight to the theory of natural selection. And this guy Thomas Hunt Morgan invented gene mapping. Not bad, 1910, and he made important advances in the study of evolution. So he took Darwin's theories of natural selection to a new level, putting a bit more of the mechanics into it. Darwin had a sort of the overall theory, but this guy was beginning to put in the practicalities, the biological side of it. The next on the list was 1935, and he was a Ukrainian biologist called Theodosius Dobzhansky. And he studied fruit flies, a very popular species to study for this sort of thing. And the method he used was recombination mapping, which is looking at the location of each trait's gene on the chromosome. So where does it appear on the chromosome? If it appears in a different place, then you've got recombination. Now this provided great evidence for genetic natural selection, and he discovered two different varieties of the same species of fly in the same area. And he found that when he examined their chromosomes, that the gene order was reversed. So something had happened, some sort of mutation, and one lot turned out to be having quite different traits from the previous lot, although they did actually come from the same bunch. And like I said, that is strong evidence for natural selection at the genetic level. Now in 1937, this Thomas Dobzhansky published a book called Genetics and the Origin of Species. Of course, The Origin of Species was Charles Darwin's book. So he was now putting them both together. So he explained evolution in terms of genes and alleles, remembering that alleles are the different versions of a particular gene that we inherit from either mother or both mother and father. Now, the three main points that he made in this book were traits can be traced to alleles and passed on to the next generation. The next one was, in a population, the variation in traits reflects the variation in alleles. So if you look at a population, you see they have these particular traits in common. Then, and you look at their DNA, you find that their alleles are also the same. And the third point was natural selection occurs when some organisms leave more offspring than others, passing on more of their alleles to the next generation. And when you think of the mathematics of it, if you start out with 2 and 2 combined with 2, then you've got 4. If 4 combined with 4, you've got 16. 16 combined with 16. Well, the mathematics are losing me there. And that can increase very, 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 very quickly. So it's natural selection is not about survival of the strongest, the ones who can knock the others on the head. So natural selection is about reproduction. It's about who produces more, who reproduces themselves more often than others. So we'll have to remind ourselves all the way through this course, we're not talking about survival of the fittest in terms of the survival of the strongest. That's a thing called social Darwinism, which was has been debunked as being a bogus theory that led to things like Nazism and a lot of capitalism, but we won't go into that. And yep, so we yep, it's all about reproduction, not about who's capable of overcoming who. And this guy Dobzhansky, in his book, established that natural selection controls which traits can get passed on from one generation to the next. And of course, as we'll find out later, it's about adapting to the environment. And the environment of planet Earth has changed a hell of a lot during evolutionary times. Not always rapid changes, a few rapid changes, but slow changes. However, one thing that has been certain is everything has changed quite often. In more ways, it can either be geological changes, climatic changes, geopolitical changes, and famines, pestilence, war, all of these things that have made life difficult. And if you had the correct characteristics or the behavioural and physical traits that enabled you to adapt to that particular environment, then you were going to reproduce more and pass on more of your DNA than someone who didn't have the necessary qualities to cope with that environment. So it was really the environment that was steering evolution. And we do have to remind ourselves quite often that there is no design in it. Evolution was not there to solve a problem. It was quite accidental. The environment changed, and accidentally, those people who happened to have the qualities needed were the ones who reproduced more. There was no one designing it. There was no one saying, how are we going to solve this problem? Well, we're going to do this, this, and this. No, it didn't work that way. And it's hard to talk without saying it as if it was designed, because our brains are hardwired for talking about things being designed. So we'll try to get into the habit of saying things have evolved, not that they were designed. They evolved in that particular environment, not to solve a particular environment. Excuse me. Sorry about that. Okay, now getting on to the modern science of evolution, which developed in the 1940s and 50s. And what developed then was a thing called population genetics. In other words, they studied large populations of people from a genetic point of view. And they were seeing how genes change geographically, in other words, through space, and over time, between generations. And this was a key to understanding evolution. And it laid the foundation for connecting genetics and evolution, which we now have. And when we get to the end of this course, you'll see why we're going into all of this, because it's actually bringing all together all what Darwin did, all of what these guys did, and putting it into, from our point of view, into behavioural, the behavioural arena rather than the physical arena. I mean, there's been a lot of work done on the physical aspects of heredity, and not a lot done on the behavioural aspects of it, which we spoke about before in one of our previous topics, was that it was inconvenient to think of human beings having instincts. It sort of just lowered us down to the level of animals, no better than animals. Well, sorry, we are animals. We're the cousins of the chimpanzees, and we both evolved from the same ancestor, whatever that ancestor might have been. We are not separate. So we just happen to have the environmental conditions that led to the sort of creatures that we are, and they didn't have it. They didn't have that environmental condition, so they remained in a certain way, and we've gone in our own way, and we'll talk more about that later. Okay, so this modern view of evolution is called the modern synthesis, or if you like, the complete title is the modern synthesis of genetics and natural selection or evolution. And it's the cornerstone of the modern science of evolution. And the four main premises are: genes are located on chromosomes and are the basis of hereditable traits. The second one is genes are passed intact from parents to their offspring. The third one is mutation and recombination are random processes that create new genetic diversity. And remembering that genetic diversity is essential for natural selection, for evolution. And we'll find out later that if a species doesn't have diversity, it will eventually go extinct. Well, not so eventually, you pretty soon go extinct. And number 4, genetic traits are beneficial and become more common over, genetic traits that are beneficial become more common over generations through natural selection. So you might say that us sitting here are the end product of the winners. We're the end product of the people who survived all the hazards that the planet Earth and the universe could throw at them. And also the hazards we're thrown at each other, that the microbiome world has thrown at us through viruses and bacteria. So we're the ones that came out on top. Or if you like, we're the ones that were stronger than all the others and bopped all the others on the head, took their stuff and went away. And here we are, which doesn't say much about us in terms of being able to live peacefully and with social justice and what do you call it, and inclusion. We'll find out more about that later. But I like a quote from the founder of evolutionary biology, or a big name in evolutionary biology back in the 1960s, a guy called E.O. Wilson, and he was at a panel discussion on socialism, and he said, socialism, a nice idea, but wrong species. In other words, to try to get a fair world with the human species is a very difficult task. And actually a course like this is a step towards doing that, because the more we know about why we do what we do, why we are the way we are as individuals and as a species, why we've constructed such social structures, and how come it is that we can't seem to make it all work. If we know why that's happening, then we're halfway to solving the problem. If you don't know why it's happening, you'll never solve the problem. You'll be throwing all sorts of silly ideas at it, because you're not addressing the real issue. And the real issue is we evolved in an environment which is very, very different from the one that we're in now. In other words, our behavioural traits were designed to survive in an environment, the evolutionary environment. They weren't designed to survive in civilization, in a civilization environment. So we've created civilization, which seemed like a good idea at the time. However, behaviorally, it's very, very difficult for us to make it work. And if you look at history in the past 10,000 years, you'll see that it's been a very rocky road, and we've never been able to completely make it smooth. We smoothed it out a little bit some places at some times, but it's never stayed that way for long. And the reason is that we are not designed for this sort of world. However, we can't go back to the old world. That's not possible. So we have to learn to make the best of a bad deal. And like I said, the only way we can do that is by understanding why we are the way we are and why we do what we do. And that's why I'm sitting here waiting for the Undertaker to come. And before he comes, I'm hoping to pass on what I've been, these crazy thoughts of mine. Okay, well, be nice talking to you, and talk to you again. Bye-bye.